of Dr. Raghunandan, BL. So, his assistant research scientist, Dr. Raghunandan, BL, is working as assistant research scientist microbiology, ASRP on biological control of crop pest on Agriculture University on Gujarat. He has completed PhD in agriculture microbiology from US GKVK Bangalore. He has worked on non-pathogenic fusarium species for the management of fusarium wilt of watermelon crops. His area of specialization, microbial control for crop pest and the disease, microbial biopesticide, microbial biostimulants. He has a research experience of 11 years. And his achievements are technology developed and recommended for the farming community for bio-intensive management of a sucking pest complex in BT cotton and also in okra. And also first report of, he has reported the first report of incidence of fall armyworm in the maize field of Gujarat, a natural occurrence of its NPV. And also he has established a biopesticide research and production laboratory with the funding of projects, RKBY funded projects on establishment of mass production laboratory of native biocontrol agent for insect pest management with outlay of 81 lakhs. An organic farming cell, government of Gujarat funded project on creation of a mass production facility of biopesticides for plant disease management with the outlay of 160 lakhs for the year 2018, 19, and 19, 20. So with this brief contribution of Dr. Raghunandan, so I request him to deliver the talk on scope of entomopathogenic, microbial biopesticide production for the sustainable organic crop pest management. So please.
Yes, sir. Sir, thank you so much, sir, for introducing me for the session. Sir, am I audible now?
Yeah, audible.
Yes, sir.
You can share your slide now.
Yes, sir. Just a second. Hello, sir. Visible na screen?
Yeah, yeah, it's visible.
Okay, thank you so much. Sir, thank you so much for introducing me for the session. And very good afternoon to all the participants, those who joined for the session, the second day of the afternoon session of the second day. Now the topic assigned to me is the microbial biopesticides production for the sustainable crop pest management. So here actually I'm going to cover in brief on biopesticides, mainly microbial biopesticide part, keeping in view the UG students, PG students, and the budding entrepreneurs. Just my presentation covers mainly the overview, the basic concepts of biopesticides, what are the production aspects we need to look in, and also the market scenario. Actually, very important component in case of biopesticides, commercialization and production aspects. So this is my outline of presentation where I'm going to cover the basics of biopesticides, of microbiopesticides and production aspects, and also followed by the market scenario of microbiopesticides will be covered. And at the end, actually, I'd like to share some of the glimpse of our laboratory where we established very recently, strengthened actually, I can say, of our laboratory in the last 2 to 3 years. So that I want to share with you at the end of my presentation. As we know, generally, as we know, the use of the pesticides, mainly chemical pesticides, certainly provide protection to the crops against the pest and diseases. And at the same time, ask a question of ourselves whether these biopesticides, chemical pesticides, are really protection, give protection. But at the same time, we need to ask ourselves: are we eating the food that is safe? But answer is certainly no, because the residue of the pesticides, mainly synthetic pesticides, are building up in food as well as in the environment, causing harmful effects to the environment, human beings, as well as non-target organisms. And also at the same time, it also affects beneficial organisms, mainly the honeybees, the pollinators. They're going to affect. If these pesticides affecting the honeybees, then definitely there is a problem for the security, food security issues. And also there are various reports citing the resistance problem to the chemical insecticides by these insect pest species with the continuous use of the chemical pesticides. So these are the issues with the chemical pesticides. So there is a need to develop a alternative measures, alternative solutions for the chemical pesticides. I think we come across this type of news in the newspapers where a lot of consignment has come back from the international market. Why? Because mainly due to the presence of the residue in the whatever the commodity we're sending. So coming to the important strategies where we look for the management of pests and diseases. As we know, the IPM is one of the important strategy. It is well established, well accepted module for the management of pests and diseases. So among the different components of IPM, the biological control is one of the key component of the IPM strategies. And nowadays, the BAPM concept is more emphasis is being given on the BAPM, that is bio-intensive pest management. The more focus is giving on the biological solutions, biological inputs for the management of pest and diseases. So as I said in the previous slides, this IPM, that is IPM strategies, the biocontrol has been proved to be an excellent alternative for the chemical pesticides for the management of pest and diseases in various agro ecosystems. And at the same time, if you look at the history of the biocontrol, so it dates back to centuries ago, 17th century, 18th century. Okay, we are working on biocontrol, we're telling biocontrol, biopesticides, biocontrol agents. But the scenario is actually, it has not reached up to the desired level, accepted level. So there are some challenges we need to address as a scientific fraternity, as a students, okay, as a different stakeholders. So we need to address some of the issues and challenges, we can say. And there is a gap in this area that we need to fulfill. If you see what are the gaps and challenges and what is the need of the hour in biological control or biological solutions in crop pest management. So we need to, first point is actually we need to reduce the heavy reliant chemical insecticides. So what's actually happening in the farm or in the farmer's field? So when any farmer notices any pest or disease, so directly will go and purchase chemical pesticides and will use. So we will not think of any other solutions, non-chemical management solutions will not never think of. Okay, and at the end, they will come to biocontrol lab or any other solutions. They will seek the solutions. So this is not the way. Actually, we need to reduce the dependency on chemical insecticides. How best we can reduce the dependency on chemical insecticides is in our hand. And also, the awareness is very, very less among farmers. So even today, they don't know the difference, the trichogramma and trichoderma. So awareness is very, very less among the farmers community on biocontrol agents and biopesticides. And also, if at all the biocontrol agents are available, but again, the availability of effective biopesticides and effective formulations is another issue that needs to be addressed. And also, even one farmer can adopt, the nearby farmers will not adopting this strategy of biocontrol biosolutions, then the strategy may not work. Okay, effectively. So that's why large scale adoption of technologies and strategies is very, very important for the biological solutions for the management of pest and diseases. Okay, and so coming to the actual topic for today's discussion, if you see the classification of biocontrol agents, where we discuss on entomophages, that is parasitoids and predators, and entomopathogens. And today I'm going to cover in detail about the basics of entomopathogens and production aspects of various entomopathogens. So these are the important components of biocontrol agents. That is fungus, bacteria, viruses, EPN, and some protozoa also. And I'm covering on fungus, bacteria, and viruses in brief about viruses also. The EPN part, I think tomorrow, that sir is covering on the EPN part. So I'm not going to touch in detail regarding the EPN part. So coming to the basics of biopesticides. Okay, basic concepts. So what are the biopesticides? So as per the classification given by the United States, that EPA, the Environmental Protection Agency. So there are three main groups of biopesticides, which comes under naturally occurring biochemicals, as we know that neem, azadirachtin is the most explored coming under this category, and microbial entomopathogens that we call microbial biopesticides, and also the plant incorporated protectants, means even the BT crop development of the transgenic crops also comes under the biopesticide part as per the classification given by the US EPA agency. So coming to the few important basics of this Beauveria bassiana. First, I'm covering about the entomopathogenic fungus, where the Beauveria bassiana, it is the most used microinsecticide, I can say. The most explored biopesticides among the fungus, entomopathogenic fungus. It is generally called by the name white muscardine fungus. Okay, and if you take the host range of the this Beauveria bassiana, it has a it is reported to infect more than 500 insect species belonging to different orders of insects, pests, and also the recent studies have been highlighting the multiple roles of Beauveria bassiana, and one of the important role you must focus on this as endophyte, because generally we use Beauveria bassiana for the control of that any pest generally, okay, by spray or some application through soil application, whatever it may be. But the colonization of Beauveria bassiana as endophyte is one of the important feature that we need to explore to maximum extent for the control of this pest and diseases. Okay, apart from the Beauveria bassiana, this metrorrhizum ansoplea also known to have this endophytic capability. So coming to the mode of action of this entomopathogenic fungus, once the spores, we can say the infective propagules, they will come in contact with the insect cuticle. So there is some enzymatic and mechanical action which leads to the proliferation of IFA inside the host body. Okay, the one important parameter which decides the efficacy of this fungus is the parameters, that temperature and humidity. Most importantly, the humidity which determines the efficacy of this entomopathogenic fungus in agroclimatic conditions. So high humidity is essential. The most essential factor is high humidity for the germination of the infective propagule, that is conidia. Okay, if once that fungus infects the insect, okay, generally establishment of the infection takes place between 1 or 2 days. It takes generally. Okay, and during the growth of the fungus in the insect body, it releases variety of metabolites. So these metabolites add on to the pathogenicity or toxicity against insect. Mortality generally, depending on the strain, depending on the host susceptibility, depending on the conditions of the agronomic conditions, mortality ranges from 3 to 5 days, but it may extend up to week also, 6 days, 7 days also. So once after the death of the insect due to the infection by this entomopathogenic fungus, okay, so there is a outgrowth of the conidia. You're going to observe outgrowth of the fungus. Generally, we're going to observe the fungal growth on the insect body. So these outgrowth actually is very important, which ensures the further spread of the pathogen in the agro ecosystem to reinfect to the another host. So this is actually in brief of how the infective propagule of the fungus going to infect the insect in ecosystem. This is an overview in the presentation of mode of action of this fungus, and also this is actually how the epizootics causes. Actually, when there is even in rainy season, in high humid conditions, generally we observe a lot of epizootics. Okay, normally natural occurrence of fungus will observe in case of ecosystem at high humidity conditions, and even rainy season also. So this is actually the representation, schematic representation of mode of action of entomopathogenic fungus. Okay, with this how spores infect, as I discussed in the previous slide. Okay, going to infect the fresh host and multiplication inside the body, producing blastospores, and also the release and reinfect the next host. So this is a representation of the epizootics transmission, epizootics occurrence. So this is actually the photographs showing the natural occurrence of entomopathogenic fungus, Beauveria bassiana, and different hosts. As I said, Beauveria bassiana, it is having a wide host range, broad spectrum of its infectivity against different insect pests. And primarily, it infects Lepidopteran at the larval stage. Apart from that, it's going to infect Coleopterans also, and some of the hemipterans also, whiteflies, thrips, aphids also. So there are some reports are there where Beauveria bassiana cause the pathogenicity against these type of insect pests. So till now, actually, we discussed how Beauveria bassiana is going to cause pathogenicity in insects. Okay, what are the infective propagules, that is spores, conidia, is going to act against insect pest. So apart from that, we need to look beyond the biocontrol. There are some agents, there are some compounds that we can explore for the other applications, other benefits also. that we can look in. Okay, actually, there are some metabolites which are produced, mainly bevericin, basinolide, basinolone. These are important metabolites which are produced during the vegetative growth of the fungus inside the insect body. So they have wide applications. They have variety of applications like antibacterial, antifungal, antiviral. Now research is nowadays is picking up in this direction, where how metabolites, how best we can utilize these metabolites for the applications in agriculture and also in the allied sectors also. Okay, so coming to the next best entomopathogenic fungus, that is Metarhizium ansoplie. Generally, we call by the Metarhizium ansoplie by the common name, that is green muscardine fungus. It represents another well-exploited fungal species after Beauveria bassiana. So it has with diverse strains are available present in the environment. And as compared to the Beauveria bassiana, Metarhizium has a narrow the host range. We're going to infect more than 200 insect species. And one more important thing, actually, we should understand, we should know this about this Metarhizium ansoplie is it is a rhizocompetent fungus. That's why actually we recommend this fungus, Metarhizium fungus, for soil-dwelling insect pests, mainly white grubs, because the rhizocompetency of the fungus is very good as compared to other entomopathogenic fungus. Okay, and very recent studies, as I discussed in case of Beauveria bassiana, how best we can explore, how best we can work on exploration of metabolites of Beauveria bassiana. In the same way, nowadays research is being going on how best we can use biopesticides for other applications, like even for salt stress alleviation, for bioremediation, and to alleviate abiotic stress in crop plants. These biopesticides have a diverse role, multiple roles that we can explore. And in the photographs, you can see the photographs what I give the petri plate, the photos of the fungus. These are actually we recorded as a native isolates of our region. We recorded. So we recorded nearly 8 to 9 different isolates from different region of Gujarat. Okay, diverse species of isolates of Metarhizium we recorded, and they belong to Ansoplie, Grunium, Major. Okay, so 3 to 4 important strains actually we documented in our Gujarat conditions. These are the photographs of natural occurrence of Metarhizium ansoplie against grasshopper, weevils, and white. Generally, as I said, these are rhizocompetent, recommended for soil-dwelling pest. It is the host strain mainly for coleopterans and for the hemipteran pest. Okay, and one thing actually I want to share with you people that one of the species of Metarhizium ansoplie, that is Metarhizium ansoplie variety acridum, it is specifically known for a biocontrol agent for locust control. Our countries witnessed the havoc attack of locusts in the last 2 years. Okay, even Gujarat side, the North Gujarat actually there was attack of locust. Okay, one particular agent, one particular product developed by in African country, that is green muzzle. Okay, it is not available in India. Okay, it is actually marketable marketed in South Africa and France. Okay, the product name, the green muzzle, it is basically made by the spores of Metarhizium ansoplie variety acridum. Okay, and the project Globulosa project in the 2002. Okay. And coming to the another entomopathogenic fungus, generally we call this Metarhizium nomoria. Now it is renamed as Metarhizium riley. Okay, generally it is slow-growing fungus as compared to Beauveria bassiana and Metarhizium. Okay, and mainly it infects noctuids because a lepidopteran species, and some species of polypter are also susceptible for this nomoria riley. And there are more than 30 species of lepidopteran insects are listed as susceptible to the Metarhizium. So this is a natural occurrence. Actually, even last 2 years back, we recorded the epizootics of nomoria riley in that invasive pest, fall armyworm. And as I said, this is a slow-growing fungus. So we need to optimize few parameters for the mass production aspect. So this is our own photograph of the rocks bottle. Okay, if you provide thin layer of the media, then you can observe the good growth and sporulation. Okay, as if you follow the method for Beauveria bassiana and Metarhizium, nomoria riley will not grow. We need to optimize few parameters so that we can explore this nomoria riley for the mass production aspects. Okay. And coming to the next best entomopathogenic fungus, that is Lecanisum lecani. It is available in the market. Okay, in different brand names. Generally, this Lecanisum lecani, the same, it is slow-growing fungus as nomoria riley. Okay, and main target pest of this entomopathogenic fungus is whitefly, mainly sucking complex. Okay, and is recommended for mainly in greenhouse condition, protected cultivation conditions where there is a control of humidity, because this requires high humidity. Among different entomopathogens fungus, Lecanisum lecani requires more humidity for its germination, for its activity. That's why it is recommended for in the greenhouse conditions or protected cultivations aspects. Okay. And mainly going to infect, it cause pathogenicity in checking complex, whiteflies, thrips, aphids, and mealybugs. And this is a natural occurrence, the photographs of Lecanis and Lecani infecting various checking complex, okay, whiteflies, scale insects, aphid also, okay, infecting natural occurrence. Main thing is high humidity is the most critical factor deciding the efficacy of Lecanis and Lecani in infecting the insect pest. So coming to one thing actually I would like to share with you people, this Iceria. Earlier actually it was named as a Pachylomyces species. I think we may remember the Pachylomyces lilacinus for nematode control. Even one product is available in India from T-Stains company. So one species, that Iceria fumicorosea, it is known to infect hemipterans as well as lepidopteran insects. Okay, and very recent invasive pest in coconut palm, that is Rogospirillum whitefly. Against this whitefly, it has been proved to be very effective. Okay, the reports are there, and product from NBR Bangalore is also being given for the demonstration purpose also, and recommendation from university also against this coconut Rogospirillum whitefly, and it is ability to cause epizootics. Okay, against this whiteflies under favorable conditions. Okay, and one product from our Multiplex Agro Private Limited from Bangalore is also they are having a Pachylomyces fumicorosea recommended for this Rogospirillum whitefly. So coming to the bacterial group of biopesticides, okay, that's BT. BT is actually it is known as acceptable green pesticide. Okay, it is not a biopesticide. Generally, it comes as a biopesticide. So we generally use it is a first green pesticide because it works on toxic metabolites. Okay, and as compared to entomopathogenic fungus like Bavaria, Metarrhizium, they have broad host range. Whereas in BT, it works very specific, very host specific. Okay, it works only on caterpillar stage. Okay, unlike fungus, it works on all the stages. Even egg stage reports are there. It's going to cause pathogenicity in egg stage of the insects. Whereas this BT works only on caterpillar stage of moths, butterflies, and maggots of mosquitoes. Okay, so important species, that is Israelensis species, is going to cause the pathogenicity in toxicity in mosquitoes also. So coming to the mode of action, actually the main factor which decides the specificity of BT against different groups of insect species is mainly by the receptors interaction. Okay, toxin and receptors which are present on the midgut epithelial cells, that interactions decides the pathogenicity, the host specificity. Okay. So, and coming to some of the basic aspects, how it looks under microscope, the BT. Okay, generally during during this growth, the Bacillus thuringiensis produce endospores. Okay, and also along with the spores, it produces some of the crystals that we call as insecticidal protein toxins, crystal proteins. That actually we explore for the development of transgenic plants also, and also these are the compounds which are responsible for causing toxicity, pathogenicity in insects. Okay, and once the cell lies, okay, after its growth, after 4 days, 5th day, okay, the cell going to lies and release the spore contents as well as this parasporal contents into the media, and this cause the toxicity. Okay. And if you see the host spectrum of delta endotoxins, generally we know this BT is going to infect lepidopterans, coleopterans, and even dipterans, okay, mosquitoes and blackflies, as we discussed in the previous slides. But apart from this, very interesting thing we should note here is it's going to infect hemipterans also, the second complex. Okay, this is very, very interesting. Very few cryotoxins, Cry2A, Cry3A, Cry11A. So these are known to infect even hemipterans also. Apart from that, in BT nowadays, research is going on for the exploration of BT for the development of against nematodes also. Okay. And one important thing is actually we should know that actually the BT is infective. Some of the BT toxins are effective against the human cancer cells also. Okay, in animal research also. So this is a host spectrum of the BT endotoxins against various insect pests in the ecosystem. And if you see the toxicity or effect of BT injection, as compared in case of fungus, we may notice the outgrowth of fungus and body becomes hardened. Whereas in case of BT, the coloration indicates it is infected by any bacteria. Mainly the color, the black color indicates the toxicity or pathogenicity by bacteria. Okay, generally, as compared to fungus, the knockdown effect of BT is very short. Within 2 to 3 days, you expect the mortality, depending on the strain and depending on the host susceptibility. And as I said, this BT is highly specific to the different species. Okay, so depending on the different strains, okay, we have different strains for different insect pests. So among these important strains, okay, BT, the variety Krustaki is the most explored. Okay, even whatever the product is available in the market, BT product, it is Krustaki based on it. Okay, as I said, for mosquitoes and black flies, it is other. Okay, these two are most explored strains, subspecies of BT among different strains of BT for the management of insect pests. Coming to one more biopesticide under bacterial group. Okay, so Bacillus populi, it is not much popular in Indian condition, but our center, Anand Center, has a legacy of report of this Bacillus populi. Okay, against white grub, the first report in India from our center. Okay, it is Bacillus populi, it is soil dwelling. Okay, bacteria responsible for the disease, milky disease, milky spore disease. We commonly call the disease as a milky spore disease in Japanese beetle, the grubs. Okay, our center reported, first report from our center against the white grub. Okay, the main constraint of this Bacillus populi is it is highly host specific and mass production is mainly constraint. It is an obligate pathogen. Okay, this is the main constraint of Bacillus populi in commercialization aspect. Coming to the third most important group of biopesticides, that is NPV. Okay, it's very important pathogen. The one group, that is Baculoviridae group, it represents the DNA viruses. Okay, the viruses belong to Baculoviridae. They are the most important pathogens of invertebrates. That is insect pest. Okay, so there are very important. There are a variety of viruses. So among different variety of viruses, the most important explored at commercial levels are NPV and even to some extent GVs. Okay, NPVs are most explored, we can say. Out of many, that too, it is highly specific, and only two more products are available as on today. Only two products, that is Liquor armature NPV and Spotted liter NPV. And one thing actually we should remember here, that the body content liquefaction is the most important identification features of NPV, and also the tree-top disease. Okay, these are two important features. Okay, as when we in the field condition, with the natural occurrence. Okay, how we want to notice the incidence of NPV. Okay, so this is regard to NPV. And coming to the mode of action, how it acts on the insect. Normally, as we discussed in case of BT, it is also a stomach poison. Okay, when the insect eats the these polylateral bodies, the polyocclusion bodies. Okay, so in the midgut, these polylateral bodies going to dissolve. Okay, because the virion particles are enclosed in a closed occlusion bodies. Okay, protein matrix. So that matrix is going to dissolve in the midgut of the insect. Okay, when in the midgut of the insect, and these virion particles or polylateral bodies, okay, they traverse through the peritrophic membrane of the midgut cells, and the infector cells, they multiply in the nucleus, and once they multiply and they come out of the cells as a budding virus, and they're going to infect the new cells. Okay, this is a mode of action. In brief, the NPV infection in insect. Okay, it is a stomach poison. And entomopathogenic nematodes, the most important, there are two important species, that is Steinernema and Ectapetes. Okay, so this actually I don't want to go in detail, because tomorrow actually we have a separate session for this nematodes. And our center, we reported 7 to 8 different isolates of Steinernema pakistanis, okay, from groundnut fields of Bhavnagar in Gujarat. The last 2 years we reported, and very last year actually we reported only one strain of this Ectapetes, okay, from our centers. Coming to production aspects, okay, what are the important things we need to look into when we go for any production aspects of microbiopesticides? So actually, before selecting any product for commercialization, mass production, we have to screen, we have to isolate, and we have to screen for the effective isolate. That is most important. Okay, so before taking up any isolate for the mass production aspects, there are series of steps, series of protocols need to be followed. Okay, we need to test against non-target organisms, storage aspects, okay, and as toxicity against other non-target organisms. Okay, what is the impact on environment? That is actually very important. Okay, and finally, patent commercialization. Apart from that, Bioefficacy point of view also very, very important. Okay, so once the strain is established, okay, from the safety point of view and toxicity point of view, so we need to establish the bioefficacy also, trials also, okay, and also develop formulations also. Okay, there is a series of steps: bioefficacy trials, toxicity, formulation, storage. So these are the things that need to be established, documented before going for any commercialization or for the mass production of microbiopesticides. So selection of species, as I said, this is the first foremost important step before going for the mass production of any microbial biopesticides. First, we need to look up. First, we have the strain. Okay, so we need to check for mortality. Okay, how best it is causing mortality in the specified any particular pest. Okay, mortality, dose rate, mode of action, what is speed of kill. So these are the important parameters we look under mortality aspects. And also, once the strain is screened under mortality, we need to go for efficiency. How best we can increase the propagule yield. Okay, what is the active component yield, and how economy point of view also. Okay, inexpensive medium used device. Okay, cost wise also we need to work on. Okay. And also a safety point of view. It must safer for the non-target organisms. These are three main aspects we must look, okay, when we selecting a species or strain for the development of microbial biopesticides. So this figure actually it explains. So when we isolate strains, okay, we'll get lot of strains in the environment. Okay, if you screen for mortality, you'll get few strains. Okay, you can see the number of arrows. Okay, and our productivity, it only get only 3 or 4 important strains. Okay, so that and at the end only we will have 2 to 3 important strains that will be feasible for the mass production aspects. Okay, this is the point actually we should keep it in mind for while taking the commercial aspects of pesticides. So as I said, first we have the strains, selection of strains and all. While we go for production process, okay, and for development, there are 2 important phases, okay, in the production of the any microbial biopesticides. Okay, depending on the pathogen, depending on the any organism, the method varies. Okay, for bacteria it varies, fungus, virus varies. Okay, we need to develop a media also. Okay, cost-effective media, quality, inoculum quality. Okay, and we need to have a control on different parameters that are going to control the mass production of important biopesticides like temperature, RH, everything. Okay, and the quality is very, very important. So if you want to produce a contamination-free or very good quality product, we should have a control on the quality aspects also. And also we at the same time, we should have a production cost, that inexpensive media, inexpensive procedure that we need to work out. And after that, once the product is ready, technical product is ready, we need to go for development of formulations, optimization of dose, application strategies we need to optimize, packaging, and finally the market. So these are the important 2 important phase for any biopesticides for the commercialization. Okay. So as I said, the product, once we harvest from the propagules, infective propagules, we call it as a technical product. Before formulation, whatever the product in the laboratory, okay, that we call as a technical product. So once you formulate it, we'll go for the production of final product for the market. Okay, as I said, depending on the pathogens, whether it is viral, fungal, or bacteria, the processes and the system vary greatly. Okay, and one important thing in production aspects, scaling up is a very challenging issue for many biocontrol companies and even for researchers also. Okay, we found many isolate strains which are very effective from the mortality point of view, safety point of view, okay, and also on the production, the cost and yield point of view also. If you want to scale up in the large scale, okay, we may not get that much effective results as compared to the small scale trials. Okay, so coming to the important technologies which we are going to adopt for the production of microbiopesticides, mainly 2 methods are generally followed. Okay, solid state and liquid state. As the name indicates, okay, first one is actually solid state means which grow on any solid support. Okay, any solid based media is used for the production of any biopesticides. And liquid state grown on the broth or liquid media. Okay, if you see the economic feasibility, okay, as I said, depending on the pathogen, the processes varies. Okay, in case of baculoviruses, as we know, it is obligate. Okay, it requires in vivo production only. It requires live host. Apart from that, baculovirus can be produced by using insect cell lines. Okay, but most accepted that method for the production of baculoviruses is the in vivo method only, the live host. If you compare bacteria and fungus, for example, bacteria BT and fungus in case of Metrium unsupplé, if you take for example, okay, in case of bacteria, it is almost always that LSF is recommended, liquid state fermentation, as compared to solid state. It can be done on solid state also, but bacteria. But the most accepted with respect to yield and although media production cost, okay, LSF is the best in case of bacteria, that is the best as thuringiensis, and in case of fungus, entomopathogenic fungus, okay, SSF is the most advisable, most important method as compared to this LSF. Even though LSF is suggested, LSF is used, but for the fungus, okay, always it is suggested, it is recommended, it is the best, that is SSF is the best as compared to LSF. Okay. And coming to, in brief, I'm going to cover what are the important methods which follow for the production of this entomopathogenic fungus. So these entomopathogenic fungus are most studied, you can say, studied widely as compared to even bacteria and other groups of entomopathogens. This group is studied widely with respect to production, optimization of production aspects, various parameters. Okay, among these entomopathogenic fungus, these two are Bacterial and Metrium unsupplé are most studied and most explored for the production aspects. So these three methods, there are three methods in case of fungus, SSF, LSF, as we discussed in the previous slide, and one important technique, you can say, there is diphasic fermentation that I'll explain in the future slides. Okay, in fungus, always, as I said, it is preferred, that is SSF is preferred over LSF. Okay, why? The reason is, I'll explain in the next slide. Okay, substrates actually generally SSF, generally simple substrates are required. That's enriched. Even without enrichment, also we get very good growth of this entomopathogenic fungus. Cereal grains. Okay, so if you use the cereal grains in the autoclavable covers, plastic covers, and trays, okay, and also various the waste of agricultural waste and byproducts, okay, that like bran and straw, okay, that can be used for the production of this, mainly the entomopathogenic fungus. Okay, and it acts as like one of the means for the conversion, bioconversion of waste into a value-added product that we can use. We can optimize different the waste of agricultural origin for the production of these pesticides. Okay, and this is how actually in our laboratory we are producing this entomopathogenic fungus, mainly Bacterial and Metrium unsupplé for the production aspects. Okay, we're using sorghum grains with enriched sorghum grains media for the production of this Metrium unsupplé, mainly by SSF method. And coming to second method, that is LSF, it is also recommended for the production of entomopathogenic fungus. It is submerged culture fermentation. Okay, we're using the liquid broth. Here actually, as compared to SSF, we're going to, here LSF, there is a production of blastospores, submerged conidia. Okay, I said no, SSF is preferred as compared to LSF. Why? Because here LSF, there will be production of blastospores more as compared to other conidia. These blastospores, they are not with respect to stability, with respect to longevity. Okay, conidospores are preferred as compared to blastospores. Even today, whatever the products available in the market, okay, biopesticide fungal based products in the market, they're all based on conidia spores, not blastospores. These blastospores produce only if you grow fungus in submerged condition, or these blastospores we generally notice in insect hemosyl. When it multiplies in the insect body, these blastospores produce. But for commercial exploration, commercial formulation, these blastospores are not that much effective as compared to conidospores. Okay. Coming to this in our laboratory, actually we developed one molasses-based media, enriched molasses-based media for the production of this Bavaria. LSF media, it holds good for Bavaria, not for Metrium. Okay. Some species of fungus, it may perform better in case of LSF, but not all the fungus. Okay. Generally, for a fungus, this SSF is preferred. Okay, we optimized by using this molasses as a basal media for the production of this Bavaria bassiana with different concentration, and also we added some additives and we optimized on media for the production of this molasses-based media. And one third important biphasic fermentation media. Okay, third important we generally follow in case of fungal-based biopesticides. Here actually both the phases are involved. Okay, first we are going to produce mycelia in liquid culture, and followed by we're going to inoculate that liquid culture in a solid-based substrate. Okay, because in liquid culture, we're going to get more of blastospores or submerged conidia. That conidia are going to inoculate into the shallow trays. So this is it is an integration of both SSF as well as LSF. As I said, conidiospores are preferred over blastospores. Here you can see the difference how conidiospores are different from blastospores. Blastospores are nothing but budding. Okay, what the spores generally they generate from the budding process. Okay, and one important thing we should keep it in mind as a entrepreneurs or scientists or students of biocontrol students. Regular passage through host insect is very essential for maintenance of virulence. Okay, so if you keep on working, keep on providing ready food for the fungus, okay, the virulence automatically, generally, gradually it comes down. Okay, in order to maintain the virulence, okay, once after 5th cycle or 6th cycle of the growth in the laboratory, we'll transfer to the live host to maintain the virulence. This is a very important thing we should keep it. So coming to the second group of entomopathogens, that is a bacterial biopesticide, it is Bacillus thuringiensis. Okay, it is easily produced in large fermenters, in liquid cultures. Okay, even at industrial level, they're using one media, that is T3 media. Okay, or actually it is very good media for even for sporulation also for BT. We're using in industrial level. Mainly there are 3 important steps in BT production. That is preparation of seeds, that is mother culture preparations, followed by fermentation and inoculation to the main tank, depending on the capacity. Okay, fermenter-based production is a well accepted production media in case of BT, and media for production is T3-based media for a Bacillus thuringiensis. And one important thing actually I would like to share with you people that how, because these biopesticides availability, as we discussed in the first slides, availability of the effective product products is a very important issue we need to address. One concept, how we can encourage the farmers to take up on-farm production. Very simple method. This on-farm production methods holds good. It works well for fungus. Okay, any entomopathogenic fungus, even for trichoderma also. Okay, even NAPHM is giving a training for exclusive training for on-farm production. How we can take up on-farm production. Okay, for the simple thing, very only sorghum-based media. One mother culture is required. For that, actually, we need to act as a facilitator, because farmers don't have any. Although it is very technical matter, okay, we can facilitate the farmers to take up the on-farm production. So the last 1 year, actually, we have been conducting this on-farm production as work, and we are promoting we are encouraging the farmers to take up on-farm production mainly for nutrition, because availability, and also if you want to register, if you want to take up the product for market, commercial product market, it takes long time. So farmers, they can parallelly, they can at least 1 or 2 times they can try for their own purpose. Okay, this on-farm production of the biopesticides, very important method. We optimized, actually we followed the NAPHM method, and we modified a few important things here for the mass production aspects, and we have been giving mother culture of 100 ml to the farmers. And one thing actually we should keep it in mind. So when the farmers are taking up biopesticides on farm level, there is no need of registration. They can take up, they can multiply, they can use for their own purpose. There is no commercial activity. If there is no commercial activity, there is no point of any registration. There is of any microbiopesticides. Okay, if the farmers are taking up the on-farm production, even they can enrich by using compost, vermicompost, whatever the already available formulation in the market, they can enrich and can use. Okay, this is one of the important method we can adopt. We can encourage the farmers to take up their farmers field. And coming to the formulations, so these are the important formulations. They are registered and CABRC, whatever the formulations in the market, mainly solid based, that is vegetable powder. Okay, and as among liquid formulations, okay, aqueous suspension is the most important. Okay. And now in future, nano formulations, they come, research is going on when formulations are developed, but market availability is, as on today, it is not. In future, definitely we may get with better efficacy as compared to the conventional formulations which are available in the market right now. So if you go, the next important point actually I would share, I would like to have market scenario. Okay, as an entrepreneur, as a scientific fraternity, we should know what is the market scenario of the biopesticides. So uh, uh, if you see perspective of biopesticides, what is the perspective? Different perspectives we can see the biopesticide in the market, depending on the source, depending on the crops, depending on the application, what are the application methods is followed, okay, and also depending on the formulations. We're going to see the formula biopesticides in the market, different biopesticides in the market. So if you see the pesticide use patterns, okay, so among different crop protections molecules used in the ecosystem, okay, insecticides occupies a major share as compared to fungicides and herbicides. Okay, if you compare the usage of biopesticides, you can say only 3 to 5% as compared to insecticide. It's stand up to 60%. Okay, and this is a huge difference between the consumption, production of biopesticides as compared to chemical insecticides. Biopesticides globally also 4 to 5%. Even India also it is around 3% of biopesticides produced as compared to chemical pesticides. Coming to the types of biopesticides, among different biopesticides, bacteria, fungus is a number of products. If you see, fungal based are more, followed by bacteria, that is BT based on only 2 or 3 important bacteria. This is Bacillus thuringiensis, Bacillus subtilis, okay, for the plant pathogen control, and virus. Okay, fungus occupies the first position with respect to number of products in the market. And in among bacteria, Pseudomonas fluorescens, Bacillus species are the major dominant biopesticide products. And in case of fungal biopesticides, Trichoderma occupies the first position, okay, followed by Beauveria bassiana, Metrrhizium, Verticillium mecani, and so on. At the global level, okay, bacteria occupies the usage types of biopesticide used. Okay, bacteria BT based products are more as compared to fungus and virus. So what are the factors we should keep it in mind when we promote the market for biopesticides? Means first of all the broad activity spectrum, because if the host range is more broad spectrum, actually the market is actually it attracts. There is a attention for the particular organism. Okay, and also consistency very important. So why there is a um, uh, awareness and also refusal from the farmers for biopesticides, because performance wise also it is we should look into the performance wise of the biopesticides, and also the production cost, low cost of the product. Actually we're discussing the production cost, production parameters. Okay, so how best we can produce with the low raw materials so that we can reduce the production cost and also ultimately reflect on the cost of the product, final product of the final price of the product in the market. And as a procedures, policies for registration, regulation aspects, this is very important. And as awareness, okay, we should encourage, we should create awareness among the farmers, these product for this pest, this disease, by pesticides. And availability, very important. Okay, even if you go and ask any insecticide dealer in the market, so they will not keep any biopesticides, even today also. Okay, availability is very important concern. Okay, in the biopesticide field, and shelf life, that is most very important. Okay, so maximum, even if you claim a scientific fraternity 6 months, 1 year, maximum 3 to 4 months only the virulence. It's by my experience. Okay, maximum 6 months we can keep the biopesticides. Okay, these are the important parameters. Okay, we have to consider, okay, while promoting any biopesticides for the commercialization or market. And coming to important, what are the major players, important companies? Actually, we should, as entrepreneurs, we should know what are the important companies that are producing these biopesticides. Biotech International, Multiplex from Bangalore, Agriland from Gujarat, and these chains. They have a plethora of products, important
There is a there is a connectivity problem from his side, so please wait. I think till he joins.
Hello, sir.
Yeah, now you can.
Sorry for the interruption. There was a connectivity issue from my side.
Okay.
Sorry, really sorry for that. And I'll continue. Just a second. Just a second. Now it's visible now, sir. Screen is visible now. Is visible now?
Visible.
Okay.
Yeah, visible.
Actually, I was at this point when the connection went off. So he prepared one compendium of commercial brand names and trade names of different microbial pesticides used in different ecosystems, where different brand names of nearly 88 companies are there in India, very important biopesticide producers. And coming to the important glimpse, actually, I would like to share our laboratory. So this is our laboratory, biocontrol research laboratory in our university, AU Anand. And very recently, we extended the biopesticide research and production unit also into the fundings of RKV and the organic farming cell project from the government of Gujarat. So coming to the legacy of our center, from 1967 is the first report from in India with regard to HANPV from Dr. R.C. Patel in 1967, and also GV, that's granulosis virus in Castor semilopa, and also RLO organisms, BT variety Galleria and white grub. Okay, so we have the legacy pertaining to biopesticide part from our center. And last 2018, we reported natural occurrence of SFNPV in fall armyworm from our center. So we generally document the important native strains of different biopesticides, microbial biopesticides. Strains, Vibrio-Bassiana, Metrrhizium, Bacillus working on biopesticide, and as Nematode, we reported 2 strains of Nematode infecting fall armyworm, and also EPN, we're recording the native strains of EPN on the biodiversity trial of ACRP. And this is a photos pertaining to the report of NPV from Anand Center. NPV in 2018 we reported, and this was a success story of our center where we adopted, where we conducted one trial in 100-hectare area in Bhavnagar area of Gujarat in collaboration with the Pidilite Industries. There are 2 NGOs which are working under the Pidilite Industries Mumbai. So we collaborated the 2 NGOs and we adopted 100 hectares of area to demonstrate mainly in the white grub management. Okay, and among the module, different components, Metrrhizium was important component, which proved very effective for the management of white grub menace in Bhavnagar area of Gujarat. And this is actually, we started small scale production of biopesticides mainly for the research and demonstration purpose only, not for any commercial activity. Okay, and very recently, actually, we established this infrastructure with financial funding from RKVY and organic cell project from government of Gujarat. We established fermenter systems for the Pseudomonas fluorescens and Bacillus thuringiensis, and we have been taken the technology from NBA, our institute, ASAP Biocontrol Project Coordinator Center, NBR. We've taken the technology for the mass production and commercialization of the technology, as there is a demand for from the farming community for Metrrhizium, Pseudomonas, and Bacillus thuringiensis. Okay, and this is a formulation unit actually we established very recently, parallelly along with the fermentation unit. And this is a laboratory actually strengthened. We established under the fundings of RKVY and organic farming cell projects under from the Gujarat. So we have R&D laboratory, inoculation facility, separate inoculation facility, and also quality control laboratory, molecular biology laboratory also. And this is activity, a glimpse of activities we generally taken up for the farmers. Mainly, we demonstrate the important biopesticides, and also we generally take up trainings for the farmers, and we train the farmers for on-farm production. Okay, so we're distributing the mother cultures to the farmers so that they can easily multiply at least 1 or 2 cycles. At least if you provide 100 ml of the product, mother cultures, they can multiply up to 5 kilos, 6 kilos of the metribuzin supply or ticotoma for their own purpose. Okay, so these activity actually we recently started in the last 1 year, and very good response from the farmers. They're coming forward to take up this activity at the farm level. So these are a glimpse of activities of our center. And at the end of our presentation, I would like to conclude my presentation with a few remarks. So as I discussed various important microbiopesticides, their market scenario, the production aspects, everything. So we need to have highly effective strains must develop and produced. Okay, and also from the production cost point of view, inexpensive production and also formulation of agent from the shelf life point of view. Okay, it must be developed, and also there is a need to encourage or need to develop the consortia of biopesticides. So as on today, no product under biopesticides registered as a consortia product. As we see in case of biofertilizers, consortia is there. Okay, NPK, bio-NPK. Whereas in case of biopesticides, no biopesticides products based on consortia that we need to develop. Okay, for that combo products, as we see in case of chemical pesticides also. Okay, and also there is a need to explore metabolites of this fungus, Entomopathogenic fungus or bacteria for commercial scales. Still, this phase is in research phase only, that metabolites part, so that we can explore for the for agriculture as well as in allied fields also that we can explore. So with these concluding remarks, I would like to thank all the participants, those who have joined, and also I'd like to special thanks to the Junior Dr. Rajay and I've been secretary of this program, and the distinguished dignitaries of the CAU Pacific College. I would like to express gratitude for the whole team of the webinar. And with this one statement, I would like to conclude my presentation. This is not an option. We are not an option by the country. It is an obligation we should follow. We have to adopt. For why? Because to save our environment, to save ourselves, it is an obligation for us, by the country. So thank you all. Thank you all and all. If any queries, you can discuss.
Thank you, Dr. Raghunandan sir. So now the topic is open for a discussion. The participants can interact with the Raghunandan sir regarding your queries, if any.
I think no queries.
I think I'll just unmute, no? Actually, there is, I have muted everyone.
Yes. Or else they can ask in the chat box.
In the chat box. Now it will go mute you on everyone. So please yourself also you have to unmute and speak. Yes. Now
Yes.
You can, yes, now you can,
yes.
as a participant, now you can ask the question.
Hello, are you getting, sir?
Yes, very good evening, sir.
Sir, it was a very nice presentation.
Thank you, sir.
Sir, I have one question regarding the identification of the different strains of fungus. Where we can be, where is these facilities are available to identify the fungus strains?
So generally, even NCL Pune actually I prefer for identification of fungus. Okay, and so do you have any right now? You have any fungal strains with you for identification, or any general question you're asking?
Sir, actually, we have in this one oil palm. There is one small rhinoceros beetle type of the one beetle is there.
Okay, sir.
We have found here in Mizoram that beetle is infected with the somewhat greenish fungus. We don't know actually what is that one.
Okay.
And we are not from the expertise from this one, microbiological control. So just for this one, we need to send this sample.
Okay, sir. Can you share the photographs of the infected samples?
Okay, sir. Sure, sir. So please share your WhatsApp number so I can send, sir.
Coordinate with you, sir. Generally,
and sir,
green fungus in coleoptile means I feel it is meteorizing.
Okay, sir.
I feel it's meteorizing.
And sir, one more question. Can you share your these slides for the benefit of this participant? If you have a problem.
I will give it to organizing secretary. Okay, you can share with all the participants, those who are interested.
Okay. Thank you very much, sir.
Thank you, sir.
Hello, sir.
Yes, tell me, please.
Sir, why biphasic fermentation is more important than SSF, or the solid state or liquid state fermentation?
Because actually, in case of biphasic fermentation, we're going to get the yield of the spore is more. Okay, if you follow the single SSF or LSF, if you compare SSF and LSF, SSF is the more important. Okay. And biphasic, why it is improvement, we can say it is a integration of two methods, and in biphasic method the spore yield is more as compared to SSF or LSF. Spore yield is more. That's why.
Hello, sir.
Yes, please.
Sir, there is a question.
Yes, please.
Sir, we will make the SSF.
Huh.
Later we can formulate in liquid.
Okay.
Is it possible?
Yeah, definitely. So whatever the products available in the market, even my laboratory, I'm doing the same thing. Okay. So just only SSF means mainly for the yield point of view. Okay. We have to get more of the infective propagates. Okay. So with the use of that sorghum or barley, whatever it may be, we get over the yield. Formulation point is second phase. SSF is only the production phase. Okay. We can formulate as such you can use. Okay. By mixing with any solid material like talc material, whatever it may be. Or else we can extract the spores. Okay. We can extract by filtration and we can make the formulation. Okay, by adding additives, liquid formulations, you can.
Sir?
Yes, please.
How to collect the solid state spores to separately, sir?
Generally, once after, generally it takes 10 to 12 days. Okay. So in my laboratory, actually, we optimized a method so that the sporulations completes in 12 days. Maximum 15 days will keep it. Okay. So become like a lumps. You have to dry the material. Okay. Under LAF or under room. What you can say. Normal condition. Okay. You have to dry the material and you have to you can sieve it. Okay, or you can use the muscle procedures we generally follow. Or else you can, instead of drying, you can add the water. Okay, in the ratio of 1:2 or 1:3, you can extract the spores. Or dry extraction or wet extraction you can follow. Generally, we follow wet extraction by using water only. 1:3 proportion. Actually, we are going to add water and we'll extract the spores and going to formulate. In case of farm level production, what we're encouraging farmers, they have to use as such. Okay, without extraction, they have to use for such in the fields, only for enriching the compost.
Dr. Raghu.
Yes, sir. Please.
This nice presentation. Just I want to ask one thing. The once we have isolated means how long we can keep the culture?
Sir,
strain how long will be virulent?
So as I said, once you isolate, okay, if you keep on culturing for 4 or 5 times, gradually decrease in the virulence is noticed.
That's why.
5 times, 5 cycles, sir, 5 or 6 cycles.
Then again we have to collect the new.
Actually, we need to charge to the live host and again re-isolate it and use it.
Okay, the same host we have to inoculate. Then after that, from that.
General host. Generally, I am following on Helicobacter parmigiana. Okay.
If we have collected from Spodoptera, it means after that 5 times, then again we have to re-inoculate.
Only, sir, actually, as I said, that this fungus is broad spectrum. You can transfer to any host. Okay.
Okay.
In case of NPV and virus, BT, it is highly specific as compared to fungus. In case of fungus, we can transfer to any host. It is pathogenic to any pest.
Okay. In every 5 cycle, we have to re-inoculate.
Generally, after 5-6 cycles, it's recommended, sir. And in case of fungus, you can use any host, provided it should cause pathogenicity or infection in the particular host.
Okay. Thank you.
Sir, good afternoon, sir. This is Kamal here, sir.
Yes, sir, please.
Sir, actually, what do you think about this consortium of Bavarian Metrrhizium? Whether it's a compatible?
Yes, compatible. Even one thing actually I would like to share with the audience that there are many products available in the market, consortia products without registration. Okay, so when we went for the preparation of this compendium, we found a lot of products with a consortia of Bavaria, Metrrhizium, Paschalomyces. Actually, I surprised when checking the details in the website and in the market also. But as of now, there is no guidelines for the registration or development of biopesticides, the entomopathogenic fungus. It's a research phase. There are a lot of studies have been proved. You can combine Bavaria with the PT, Bavaria with Metrrhizium and supply. But no guidelines as of now for the registration and the use of. We can suggest the farmers tank mix. Okay, so they can procure separately. While using, they can mix it and spray, or they can use tank mix. We can suggest.
Right. Thank you.
It is advisable. Consortia product actually we can advise, but we can definitely take in near future. Products may come without registration already available in the market, but off the record. Okay.
Hello.
Yes, please.
Sir, I have tried the Bavaria bassiana against the rice stem borer.
Okay.
No insecticide is not affected against it. All are resistant.
So I'm not getting. Please, can you repeat?
Sir, I have used all the chemical pesticides and biopesticides separately. All chemical insecticide has failed against the rice stem borer.
Rice stem borer. Is it a lab trial or field trial?
Sir, I have strain from the Pune.
Okay.
I developed it in the liquid stage. I have sprayed on the 500 acres, sir. I tested.
500 acres you tested?
Yes, sir.
So what I am asking, that's why any preliminary study have you conducted because we are going for 500 acres of this Bavaria bassiana or any other chemical insecticides. So we should have some recommendations or preliminary study, okay, for large scale use. Na?
Sir, from 1.5 years to 2 years, I am working on Bavaria bassiana only, sir. Okay. That's why I have tried this.
Okay.
Farmers have.
You are saying Bavaria bassiana is not effective against stem borer?
It's effective, sir.
Okay. Okay. Yeah, that is a good point. No? If it is effective,
it is effective more than the chemical insecticides.
But stem borer, at what stage actually you are using the Bavaria bassiana? Okay. Yes.
At larval stage only, sir.
Okay. But we need to check it.
60 to 75 days period.
Okay.
Of rice.
Application method, spraying.
Spraying, sir. Liquid spray.
Spraying. Okay. Any other question, please?
Sir, which strain will work on the brown plant hopper, sir, on rice?
Hoppers mainly, I suggest, and my experience, Metrrhizium is the best. Okay, for hemipterans, always I suggest and recommend the Metrrhizium. Okay, we have one large scale trial, and even we had trials on mango hoppers, Metrrhizium and supply. We got very good results. Okay, even this actually we are going for 500 hectares demonstration in mango. In Gujarat, actually in coastal area, the complete mango belt. We are going for demonstrate Metrrhizium against mango hoppers. Metrrhizium I can suggest for the hoppers.
Any other questions from the participants? They can interact.
Okay, some questions in the chat box. I think it's over, no? Yeah, from chat box.
We discussed, sir. We discussed it.
Okay. All right. So finally, I would like to thank Dr. Raghunandan, BL Assistant Research Scientist, Microbiology, ASRP on biocontrol of crop pest on an Agriculture University on Gujarat. Thank you so much, sir, for your valuable time and delivering the talk on the microbial biopesticide production for the sustainable organic crop pest management. Thank you for accepting our invitation also for presenting, and it was very informative, and I hope all the participants they could even interact at the end. There was a very fruitful discussion at the end. Thank you so much, sir. Thank you.
Thank you so much, sir. Thank you so much.
So tomorrow we will have a final technical session from morning and the afternoon. Please, everyone, please join. And I will share all these YouTube link also. We will send it tomorrow. Thank you.